Okay, so if you follow groundbreaking science, you've definitely heard about CRISPR. It's everywhere. They
Absolutely.
call it molecular scissors, right? Because it's this, well, revolutionary gene editing tool.
Exactly. It holds the potential to not just manage genetic diseases, but maybe even cure them. At the source.
Which is incredible. And it came from bacteria, like a defense mechanism.
Yeah, that's right. And scientists figured out how to harness it, essentially to precisely cut and paste DNA sequences.
Amazing. So today, our mission is really to dive deep into that promise, the medical side, but also the big ethical questions that, inevitably come up.
Definitely. It's a huge topic on both fronts.
So let's start with the impact. When we talk about tools like CRISPR, where is it actually, making a difference now?
Well, it's already happening. It's not just theory anymore. We're seeing a fundamental shift moving beyond just managing symptoms.
To actually fixing the underlying problem.
Precisely. Rewriting the faulty code, so to speak. The big example right now is Casgevy. It's the first FDA-approved CRISPR therapy.
Okay, Casgevy, what does it do?
So it tackles sickle cell disease and also transfusion-dependent beta thalassemia. What it does is they take a patient's own blood stem cells.
Outside the body.
Yes, edit them to basically switch on a healthy fetal hemoglobin gene and then put them back.
And that addresses the root cause of these blood disorders.
Exactly. And the results have been, well, pretty remarkable. Truly life-changing for patients.
That's fantastic. But I guess with that kind of precision, the research must be exploding in other areas, too.
Oh, absolutely. The potential applications are just incredibly wide-ranging. Think cancer therapy, for instance.
How does it apply there?
Well, researchers are exploring using CRISPR to engineer a patient's own immune cells, basically teaching them to better target and destroy cancer cells.
Wow. Okay. What else?
Then you have neurological disorders. There's active research looking into correcting genetic mutations linked to Huntington's, Alzheimer's, Parkinson's. The list goes on.
Complex conditions.
Yeah, very. And infectious diseases, too, like HIV. The idea is maybe using CRISPR to directly disable the viral DNA within our own cells.
That would be a game changer.
Completely.
Yeah.
And then think about all the rare genetic diseases. Thousands of them. Many with no treatment options at all.
Right.
CRISPR offers this unprecedented hope of creating bespoke therapies, tailor-made fixes for specific, even unique mutations. We actually saw a case,
Oh, yeah.
a groundbreaking one, for a baby with a life-threatening genetic disorder. They essentially crafted a unique therapy just for that child.
That's incredible. But it also sounds incredibly powerful, which naturally brings us to the other side of the coin, the ethics.
Yes. And it's crucial we talk about this. Wielding this kind of power over life's blueprint, it demands very careful consideration.
So what are the main concerns? Where are the big ethical questions?
Well, first off, there's safety, off-target effects. CRISPR is precise, but it's not perfect.
Meaning it could cut the wrong place.
It could make unintended edits, yes, or sometimes it might not edit all the target cells successfully. You get this mosaicism where some cells are edited and some aren't.
Which could affect how well the therapy works, I imagine.
Exactly. So ensuring long-term safety, predictability, that's absolutely paramount. We need to know the edits are consistent and reliable.
Okay, safety first. What else?
Then there's a really critical distinction we need to make. It's between somatic gene editing and germline gene editing.
Explain that.
Sure. Somatic editing affects only the body cells of the person being treated. So those changes aren't inherited. And that's becoming more accepted, especially for treating serious diseases.
Like the Casgevy example.
Right. But germline editing, that's different. That involves altering DNA in sperm, eggs, or embryos.
Meaning the changes could be passed down to future generations.
Precisely. And that raises profound ethical alarms. Unintended consequences for the entire human gene pool, the whole idea of, quote, designer babies. It's a very serious line to consider crossing.
A line with huge implications. So beyond the individual patient or even future generations, what about society as a whole, this power? What does it mean for us collectively?
That brings up equity and access. It's a major concern. Will these CRISPR therapies be incredibly expensive?
Yeah, I was wondering about that.
If they are, we could end up creating a kind of genetically privileged class, people who can afford cures or enhancements while others can't. That's a huge ethical challenge. How do we ensure access for everyone?
A massive question.
And tied into that is the classic slippery slope argument. Where do we draw the line between treatment and enhancement?
Right. Fixing a disease versus, what? Making someone smarter,
Stronger, exactly.
Mm.
If we can correct genes for diseases, what stops us from using CRISPR to, say, boost intelligence or change physical appearance? It forces us to ask really fundamental questions about what it means to be human.
And potentially leads to less diversity.
Potentially, yes. And one more point on germline, specifically informed consent. Future generations who would inherit these changes, they can't consent. That's a huge ethical hurdle.
Wow, okay. This has been really illuminating. So much potential, but so much to carefully consider. As we wrap up, what's the final thought here?
CRISPR's future looks undeniably bright, right?
It does. The potential to eradicate disease, to alleviate suffering. It's immense, truly immense.
But harnessing it responsibly for the good of all humanity, that takes more than just brilliant science. It demands this deep, ongoing, global ethical conversation. We have to proceed with caution and wisdom.
So thinking about everything we've discussed, this power to edit the very blueprint of life, it leaves you wondering, doesn't it? How might CRISPR ultimately redefine what it even means to be human?